Diabetes is a collection of disorders which is characterized by a defective regulation of carbohydrates, fats, and proteins. Here, the chronic hyperglycemia is basically the biochemical hallmark of type 2 diabetes, which is characterized by a absolute or relative deficiency of insulin. The clinical manifestations of diabetes ranges from the asymptomatic diabetes to the life-threatening diabetic ketoacidosis or hyperosmolar ketosis to death as well. The life expectations. Type 2 diabetes represents an enormous, rapidly expanding global public health problem, wherein the life expectancy is dramatically reduced to 10 to 15 years if it is diagnosed in the middle age. Further, the quality of life of diabetic patients is also drastically reduced. Pathological hyperglycemia sustained over several years leads to a structural and functional changes within the tissues, which makes the microvascular complications irreversible. 70% of the causes is attributable to the macrovascular complications like ischemic heart disease, cerebrovascular disease, and the peripheral vascular disease. There is a reduced life expectancy by almost 10 to 15 years if the diabetic patients are diagnosed at the middle age, and also there is a reduced quality of life of the diabetic patient, which is drastically hampered. Let us understand the role of insulin in glucose metabolism. Insulin is a principal anabolic hormone which exerts its myriad action on the intermediary metabolism, ion transport, and the gene expression. Insulin is secreted from the beta cells of islet of Langerhans of the pancreas, and the metabolic actions of insulin is antagonized by the counter-regulatory hormones like the glucagon, catecholamines, and the growth hormones as well. Physiological action of insulin is broadly classified into two types. First is the metabolic effect and second is the long-term effect. The metabolic effects are generally the acute effects or the short-term effects. The metabolic effect of insulin includes the suppression of HGO, that is the hepatic glucose output. Secondly, the insulin is also responsible for the promotion of glucose storage as glycogen. Thirdly, the insulin is also responsible for the insulin mediated glucose uptake or disposal into the peripheral tissues, or maybe the hepatic tissues, or maybe the adipose tissues. Furthermore, insulin is also responsible for the suppression of lipolysis and ketogenesis, wherein the long-term effects of the inhibition of insulin is very tightly matched with the circulating glucose concentrations. The insulin is secreted at a low background level throughout the day, which accounts for approximately 50% of the total insulin secreted. The remainder amount of the insulin is secreted in close temporal association to the rise in the portal plasma glucose following the meals. So the glucose lowering effects of insulin primarily results from two important factors: the suppression of HGO, that is hepatic glucose output, and second is the stimulation of the insulin mediated glucose output. Understand what is hepatic glucose output. As the name suggests, there is an output of the release of glucose from the liver. It happens by two ways. The first is the glycogenolysis, which means the breakdown of glycogen to form glucose molecules, and second is the gluconeogenesis, that is the formation of glucose from the new source, that is the non-carbohydrate sources like the proteins and the lipids. Glucose lowering effects of insulin is not only results from the suppression of hepatic glucose output, but also the stimulation of the insulin mediated glucose uptake into the skeletal muscles, adipocytes, as well as the hepatocytes. Now let us understand the entire process in brief. Once the insulin molecule has docked onto the surface of the insulin receptor, that is the alpha subunit, it leads to a series or the cascade of post signaling events. There is a autophosphorylation of the beta subunit, that is at the tyrosine sites. Now substrate for the insulin receptors are the insulin receptor substrate 1, that is also called as the IRS1. Further phosphorylation and the dephosphorylation events eventually leads to the activation of enzyme like the glycogen synthase. Now this insulin receptor substrate 1 is also responsible for the expression of certain genes, ion transport, the lipid and protein synthesis, the activation of PI3 kinase, that is the phosphatidyl inositol 3 kinase, which ultimately leads to the translocation of the GLUT4, that is the glucose transporter 4, from the Golgi bodies to the cell membranes, wherein they uptake or absorb the glucose molecules from the extracellular fluid to the inside of the cell. The uptake of these glucose molecules inside the cell, they undergo metabolism and finally yield ATP, and they are stored inside the mitochondria. Types of glucose transporters like the GLUT4, which are present over the surface of the skeletal muscles, adipose tissues, and the hepatocytes, whereas the GLUT2 is especially present on the islet of the pancreatic beta cells, and it does not require the presence of insulin for the glucose to get entry inside the cell. Whereas the other isoform of the glucose transporter, which is also called as the GLUT1, it is present basically in the blood brain and the blood retinal cells. Pathophysiological determinants of type 2 diabetes mellitus remains the defective insulin secretion or the impaired insulin secretion, and second is the insulin resistance. Now what exactly is the defective insulin secretion? Type 2 diabetes secrete enough insulin to prevent ketosis but insufficient insulin to maintain the normal blood glucose concentrations. A 30 to 40% reduction in the insulin mediated glucose uptake further leads to progressive compensatory fasting hyperinsulinemia until the fasting plasma glucose levels exceeds 7 millimole per liter or 126 milligram per deciliter. Then thereafter what happens? The endogenous production of the insulin progressively declines. This leads to the deposition of the islet amyloid polypeptide inside the islet cells of Langerhans, thereby leading to the beta cell dysfunction. Understand the second most important pathophysiological determinant of type 2 diabetes mellitus. This is called as the insulin resistance. Insulin resistance is defined as a reduced biological response towards a physiological amount of insulin. The number of insulin receptors goes down, or there could be a reduced affinity towards insulin, which is also called as the down regulation of insulin receptors. Insulin resistance per se is asymptomatic, and this is one of the most important metabolic feature of type 2 diabetes mellitus. There is a fasting hyperinsulinemia in the presence of normal or elevated blood glucose levels. With this, I conclude this video. In the next video, I will be speaking on the risk factors of type 2 diabetes and the metabolic syndrome. Thanks for watching and listening this video. Have a great day ahead.